looking forward to your talk.
Good afternoon, everyone, and thanks for being here. Today I would like to share with you a slightly different way to look at sex change. I've been working on this project for a few years now in collaboration with two colleagues from the University of Salford, Dr. Ilaria Coscia and Professor Stefano Mariani. So sex change is a striking phenomenon in the evolution of mating systems, and to me is the most remarkable example of sexual plasticity. It has been described in a variety of animals, mainly invertebrates, a lot of crustaceans and mollusks. And among vertebrates, this is peculiar to teleost fish. And it has been described in over 450 species belonging to 30 families. If we look at sex change has evolved in fish multiple times. And if we look at the phylogenetic tree, of course, this is just a very simplified example, we can see that the majority of species are female first sex changers. So they are protogynous species. So they start their life as females and then they become large dominant males. And a fewer number of species are protandrous. They are male first sex changers. And in this case, they start their life and reproduce as male, and then they change to large, more fecund females later on in life. There are also a few species, 22, that can change back and forth to the two sexes. So we do have a good understanding of the ultimate causes of sex change. Since the size advantage model was proposed in 1975, we think we understand that it is sex change is advantageous when the reproductive value of each individual depends on the size, but more so for one sex than the other sex. So this relationship is different for the two sexes. So we can have situations where if males are having the same reproductive success, independent of their size, it might be better to be a small male first and then change to a larger female later in life, because larger females can produce more eggs. But if only larger males are really successful in reproduction, then it might be better to be a small female first and then change to large males later in life. So this is from the individual perspective. So individuals are maximizing their fitness, becoming large males or large females. And, but this can be scaled at the population level. So we can look at the lifetime variance in reproductive success, which is the lifetime variance in the number of offspring that each parent can produce. And this will have an effect at the population level. And indeed, some study has been looking at the population genetics in sex-changing species, but most of the time the focus has been mainly on protogynous species or comparing gonochoristic species, the one with fixed sexes, with sex changers. To us, this is a little bit of a limitation because protogyny and protandry are quite different strategies. So indeed, both of them start with a lot of small individual of the first sex that become larger individual of the second sex later in life, but the reproductive mode is different. In protogyny, we have haremic species or maybe large spawning groups, while in protandry, there is near random mating or monogamous matings. Also, the variance in reproductive success is expected to be high for the second sex in both strategies, but more so in protogyny, because here if we have a harem, very few males will be able to dominate the harem. And finally, the sex ratio is unbalanced towards the first sex in both systems, but again, more so in protogyny, as demonstrated in general for sex-changing animals by also Pence-West in 2004. So if we look at sex ratios and individual fitness, these two parameters are key to estimate effective population size. Effective population size, NE, is a critical value not only in population genetics, but also in conservation biology, because it gives us an example of the intensity of the genetic drift of populations and the genetic variability. And So given the effective population size, we might have an idea of the resilience of population to environmental changes. So because the variance in reproductive success is so high in protogynous species, and because the sex ratios reinforce this variance, we are actually expecting a lower effective population size in protogyny than in protandry. And this is our working hypothesis. We are expecting that the direction of sex change is going to have an effect on the effective population size of a species. To test this hypothesis, we chose 8 sex-changing species. 4 of them on top here are protandrous, and 4 of them are protogynous. And for the rest of the presentation, the protandrous species are always in blue, and the protogynous species are always in red. So for these 8 sex-changing fish, we estimated effective population sizes with two independent approaches. Initially, we calculated life history tables, and we using data from FishBase and from primary literature, and we use these life history tables in LDNe, a software that can estimate effective population size. And then for 5 spared species, we also add molecular data. So we use microsatellite data to estimate effective population size with the linkage disequilibrium using the software LDNe. So this is an example of the life history tables. As you can see, we have maximum age, maximum length, and various parameters for fecundity and growth. And these are the results from LDNe. I'm not expecting you to read the table, but we did calculate census size, adult number of individual in population, generation length, variance in reproductive success, and then effective population size and effective number of breeders per breeding cycle. And if we look at the results, again with the protogynous species in red and the protandrous species in blue, we can see that there was no significant difference in census size, actually in adult size, the number of adults in the population, but we did, so no significant difference there, but we did find higher variance in reproductive success in protogynous species, which resulted in lower effective population size, as we were expecting. Also, the number of breeders was lower in protogyny than in protandry. So looking at the life history tables, it seems that our hypothesis was holding. We went a step further, and we looked at average number of offspring, VK and B, for each sex. And here, if we look at the results, and if we look at the first sex versus the second sex, so females in protogynous species and males in protandrous species versus the second sex, we can see that both systems has a similar strategy. So in both systems, the second sex is the one where there is a higher number of offspring and a higher VK, and a lower number of breeders. So it's the same strategy, but the magnitude is actually higher in the protogynous species. And we also look at the interaction between the first and second sex and the mating system, protandry and protogyny, and we can see that in general for K and VK, in both systems, protogyny is doing better than protandry. For the first sex, a little is the opposite, but the difference is really minor, and the inverse trend for the number of breeders. At this point, we looked at the molecular data, and when we calculated effective population size for different species of 5 sex-changing species, and for the protandrous species, we did have very high values, up to infinite numbers of effective population size. But when we look at the protogynous species, one species was actually behaving according to our prediction, not so much the second one, Pagellus erythrinus. Pagellus erythrinus, though, is not an aramic species. These are a group spawner. So we were lucky enough to have 2 more data sets from literature, 2 groupers, 2 protogynous groupers. One of them is aramic, and actually effective population size in these clusters with Chrysoblephur puniceus, the other aramic species we had. And the second one, Ephenephilus guttatus, is a group spawners. So indeed, we can recognize that the Protandrous species, which are characterized by random mating, have a higher effective population size than the Protogynous arenic species, and the group spawners are somewhat in between in terms of effective population size. So we have a gradient that it's driven not just by the system, but also by the specific reproductive strategies. So this is another way to visualize it, actually taking care of considering one over effective population size, because working with infinite values is a little tricky. So if we have 1 over infinite, we have 0, and it's easier to visualize it, and again, to see the difference between these different groups. So I hope that I showed you that the direction of sex change makes a difference to estimate effective population size. We indeed have a much lower effective population size for Protogynous than for Protandrous species. And so clustering all of these sex changers in one homogeneous group might be a little misleading for conservation and management of stocks. And many of these sex-changing species are indeed commercially important. Also, when we looked at the molecular data, we realized we discovered an extra layer of complexity. I am a behavioral ecologist. I always think that it's really important to understand the reproductive strategies, the behavioral responsive of species. And indeed here, we did find an extra layer of complexity when looking at the mating strategies, with Protogynous species behaving differently if they are aramic or if they are group spawners. So with this, I would like to thank all the people involved in this project, especially the one that helped me with the molecular data, giving us the molecular data, or helping me for the sampling of Diplodocus sarcus, which was the species I mainly worked on, including the people that helped me to get rid of the dead bodies at the end of the collection. And also I would like to thank you all for your attention, and I'll be happy to take any questions if you have any. Thank you.
Fascinating system, nice data. Other questions? Okay, so I have a question.
Yeah.
How is it the same environmental effects that induces sex change from male to female and from female to male? Or are these different factors?
So there are different, actually for Protandria and Protogynous, often the triggers are different. In some species it's more environmental, and some species is more social. So especially for Protogynous species, when you have a large male that is dominating a harem of females, if you remove that large male, the bigger female will change to male. So it seems that there is, the larger males are kind of restraining the females into changing sex. This is not the case in Protandrous species. In that case, it seems that reaching a certain size, they will switch to becoming females. So sometimes it's social, and sometimes it's environmental, and sometimes we don't know exactly how it works. Yeah.
Any question? Yes, please.
Is there a latitudinal gradient with like the ability to sex change? Is it something that sort of, I don't know whether I'm wrong in thinking this, I associate with tropical ecosystems?
Yeah, we are actually working, we have now a large data set. We are trying to plot the temperature, latitude with sex change. It appears that sex change is in general more common in warmer waters, in tropical waters, and closer to the coast. So there are environmental variables also, and this is something that with our group are working at the moment. So I might be able to tell you more soon, hopefully.
Okay, we have to continue. Thank you very much again.
Thank you.
And we come to our next speaker, who is Nina Gerber from the University of Zurich. And this is your talk. 1 second. Stage is yours.
Thanks. So I will talk about sex and dispersal in space and time. And as you might know, in evolutionary biology, the prevalence of sex is a key question because compared to asexual reproduction, sex is very costly. And one way how sex can be stabilized is when there are benefits. So the costs are reduced and there are net benefits to sexual reproduction. And one way such benefits can be achieved is by the link with an ecological function. So for example, in cyclical parthenogens, where organisms can reproduce sexually or asexually, and resting eggs to survive winter can only be produced through sexual reproduction, then of course sex is stabilized. And we ask here, why should such a link between sexual reproduction and dispersal in space or time evolve in the first place? And I have to disappoint you here, I don't have the answer yet. But I'll present very preliminary results and we'll see what happens. So organisms live in a changing world, and the world they can change over space. So there are habitats that are of higher quality or where individuals are better adapted and they face different challenges. So they can be predators, for example. Different habitats can have different microclimates, or there can be pathogens, and environments do not only change in space, but they can also change over time. So at the next time step, things can look very differently. And this is very challenging, and there are several ways how individuals can cope with that. And one way is adaptation. So individuals can adapt when they're in bad environments. And I'm not talking about plasticity here, but I'm talking about they can produce offspring that are better adapted than they are themselves. And for that, sex is very helpful, because if you want to have offspring that are different from you, asexual reproduction usually doesn't really help. Another way how they can cope with changing environments is they can disperse in time. So if you're in a bad environment, just disperse, and hopefully some of your offspring should disperse, and hopefully some of them will end up in an environment where they are better adapted. A third option would be time. So you produce dormant offspring that can then endure bad conditions, and when they wake up again, maybe they are in better conditions. You can also do nothing and your offspring stays, but this option is probably not favored by natural selection, so we'll just ignore that for my talk. So there are these three options, how they can escape bad conditions. But if there are those options and there is a need to escape bad conditions, why shouldn't they invest in all means to do so? So why shouldn't they produce dormant seeds that are produced sexually and that disperse? And the question to this lays in bed hatching theory. So what all those strategies have in common is that there is uncertainty about the environment where the offspring might end up. And a bed hatching strategy is a strategy that spreads the risk. And I'll show you this as an example with the dispersal. So if you imagine a strategy where no one disperses and everyone stays in the same pond when there are bad conditions, what you can see on the left side is conditions stay bad, everyone dies. So you have a fitness of 0, or then the environment changes and everyone survives. So either all or nothing is what you get. But when you include dispersal and some of your offspring disperse, then a proportion of your offspring might end up in a habitat where they are well adapted to, and thus a proportion survives, and it's not this all or nothing. And if we track the performance of those two strategies, everyone does the same thing, and the bed hatching strategy over several time steps, then we can see, well, here for 10, then we can see in one strategy there's always everyone or no one, and in the other one there's a proportion. And if we have a look at the mean and the variance in the fitness of those strategy, we can see that in the bed hatching strategy, in this case dispersal, the mean is reduced because dispersal obviously has some costs. Not everyone that disperses might end up somewhere. There are challenges in dispersing, but also the variance is reduced. And everything else being equal, natural selection will favor strategies that reduce temporal variance. So if the environment is unpredictable, bed hatching might pay, or is a strategy that is selected for. So Now, why not disperse in space and time and have sex at the same time? And the answer to this is sex and dispersal in space and dispersal in time can be seen as bed hatching strategies. So, and if one of those strategies evolved to minimize the variance, then for the other ones there is no scope left to further minimize the variance, and what is left are costs that are added to it. So those three strategies shouldn't evolve. But we actually find this link between dispersal in space and time and sex in many systems, as for example, as I already mentioned before, cyclical parthenogens as aphids, rotifers, or daphnia that can only produce resting eggs via sex, or this association is also found in plants or fungi that produce spores or seeds that disperse, and those spores are produced sexually. So why do we find this link? And here we try to find conditions when this link co-evolves. And to do so, we created a model, an individual-based model, where the individuals, they live in a world that can look like that, or which is pretty smooth, and is temporally not variable, or they can also, we can change the world, and they are actually in a world that look like that, and it would also change over time, but here I'm actually not sure now where I can start the video, so it's just a huge mess. And the world is changing in space and over time, and we can have different autocorrelations in the world. And then we will get there. So I may continue with talking, but it will be back, and then you'll see what happened in the meantime.
You can wait.
Well, that's fine. I can just talk.
But why is this shit not working?
Great. Now I just need to
Yeah.
go backwards. Great.
Sorry.
Okay, so to come back, in this world there are individuals, and those individuals, they have several traits. So they have an environmental matching trait, and they have a location. The location tells us where in the world they are, and we can see what the environment has, what kind of environment they're in there. And with those two information, we can calculate how adapted they are to the current environment where they live in, which gives us the maladaptedness, which is really low here. And the maladaptedness, what is important, what you need to remember throughout the talk, is a maladaptedness of 0 means they are really good adapted to where they are perfectly adapted, and high values of maladapted is really bad for them. And then in addition to this, they have a reaction norm for sex, dispersal, and dormancy, which look the following. So on the x-axis is how well adapted they are, and on the y-axis is a probability to do something like that. And the shape of this curve is defined by 4 parameters, which you don't need to remember, but it's just important that those are the traits that will evolve. And with that parameters, then the curve can take any shape. It can be very flat, it can be reversed, or the midpoint can be somewhere. And so we have our individuals, and they have a reaction norm. Depending on how well they are adapted, they react differently, and they have a higher probability of having sex, disperse, or go dormant for their offspring. So they are somewhere in the world, then they can breed, and what is also important is the better adapted they are, the more offspring they can have, so that's where selection kicks in. And according to their reaction norm, they produce asexual or sexual, and then they disperse according to their reaction norm of dispersal. They go somewhere, some of them might die because dispersal is costly. Then they go dormant, again, some of them can die. And in the next step, the world might change because we go further in time, the world changes a bit, and then the offspring can hatch from the dormant eggs and everything starts from anew. And we can track the evolution of those parameters that I've just described before, and we can see what happens over time. So here you can see the evolution of A, B, C, and D, which define the shape of the curve. The only thing that is important from this graph is that you can see that things converge quite nicely. What you can see in those extra bits, right to the right side of the graph, is the individual variation. So this is really, really small, and the rest is population means. So we can see that things converge quite nicely. If we then see what happens in a world where there is no temporal variation, in the middle, you can see that they are quite nicely adapted to where they currently are. The maladaptedness is really low. But what we see when we look at the reaction norm then is sex is really low, so that was kind of expected. Dormancy is also quite low, but dispersal is really high. Also, when we look at the graph on the very right side, we can see that everywhere in the world, the proportion of disperses is really high. And what we also see is that the curves are really flat, so they don't really react to how well adapted they are. But here we also don't have any temporal variation. So let's have a look what happens in environments that vary over time. Vary more over time, and even can become unsuitable at some time steps, what is indicated here by the black hole in the graph. So here individuals are not that well adapted anymore. And now let's see what happens with the reaction norm. Again, dispersal, which is this curve here, the utmost curve, it's really high. And then there's dormancy. Now it's a bit higher than before because they can endure unsuitable times. And sex is really low, and still the curves are really flat. And if we increase the costs of dormancy and dispersal, we can see that still the curves are really flat, but they are just lower. So the proportion of sex is a bit higher, but the curves are lower. But we find something pretty interesting here, what is that we have high dispersal when they are very well adapted. So if you look at the left side, where they are well adapted, dispersal probability is high. And on that side, it's even more extreme. And then we were wondering, how can this happen? Why should they disperse more when they are well adapted? And that's where I would like to change the title of my talk to The Evolution of Dispersal Through Kin Competition, because this is actually what happens here. So when they are very well adapted, kin competition is really, really high in that system because because there are many offspring from the same parents, and they compete, and then it still pays to disperse, even though you're very well adapted. And with this, I would like to conclude that kin competition can promote dispersal. We found, to come back to the actual question that we asked in the beginning, that sexual reproduction is generally low, and it increases when the costs of dispersal and dormancy increase, but still it's really low, and that the reaction norm that evolved are really flat, which is pretty puzzling, so they don't really react to how well adapted they are, even when the costs are high. And what we can conclude is that we need to explore this a bit more, and more work needs to be done. And with this, I would like to thank the coconuts, my group, and you for your attention.
Thank you very much, Nina. Other questions? So then I have a question.
Yeah.
Are you also doing instrumental? I mean, the pure model, which is really fascinating. Are you somehow boosting this up with experiments or with field data?
So not for this one, or not yet. So this is purely theoretical. But we do work with Daphnia, which you can see, to support models. But we need to see where it goes, and we need to have clear predictions to see how we could test them experimentally or what could be done. But this work was completely based on simulations.
Yeah, and probably simulating dispersal is a bit difficult with Daphnia in the lab.
Well, in the lab, yeah, of course. Yeah.
Okay, any more questions? This is not the case. Thank you very much.
Thanks.
And we come to our next speaker, who is Nicola Petkovic from the University of Edinburgh. And you proceed. Fine.
Hello everyone, my name is Nikola, coming from the University of Edinburgh. I'm a third-year PhD student, and here I'm going to talk about an experiment that has been recently performed about evolutionary rescue in environment deteriorating in a complex manner. So evolutionary rescue is the process of adaptation to conditions lethal to ancestors. The population subjected to the stressful environment is maladapted. It has a negative growth rate and the population density falls. If it falls below the critical threshold, which is a critical low population size, it becomes highly vulnerable to extinction due to demographic and environmental stochasticity. For example, deleterious alleles may become fixed. The only chance for survival is evolutionary resistant genotype by mutation and its rise in frequency above the critical threshold. There are plenty of factors that can influence the likelihood for evolutionary rescue, and I'm especially interested in two of them, which are sex and biotic interactions. There are several explanations how can sex be beneficial when conditions change, and most of them are based on the fact that sex increases genetic variance available for selection. For example, if there are two or more beneficial mutations independently arising in different individuals, sex enables faster assembly of these mutations in a single individual. Sex also prevents accumulation of deleterious mutations by generating genotypes least loaded with deleterious mutations. Combinations of allele on a different loci may be underrepresented in a population which is termed negative linkage disequilibrium. Recombination breaks this equilibrium and increases the frequency of these beneficial combinations. So for all of these effects, sex makes selection more efficient and enhances adaptation. A growing body of data has provided the support for this theoretical framework, and many of these studies have been performed on Chlamydomonas stringhatiae. It is a unicellular alga, very often used as a model organism because of its fast growth rate, and particularly interested because it can reproduce both asexually and sexually, which can be easily manipulated in laboratory conditions. So this alga has been subjected to various types of a gradually deteriorating environment, and some sexual populations have evolved stable growth in extreme conditions, such as obligate heterotrophic growth in light absence or in high salt, which concentration exceeds the marine conditions. So these studies suggest that sex can not only rescue from extinction, but it can also but it can also extend an ecological niche of an organism and trigger evolution of new ways of life. So these studies suggest that sex can be beneficial in environment deteriorating in a simple way, but which involves one component of environment that changes. It is still uncertain, will it prevent extinction if the change is complex? And here by complex change, I mean the change in abiotic and biotic component of environment. Incorporating another species in the study of evolutionary rescue is important because in nature each species is interconnected with the biotic component of its environment through multitude of interactions which affect its survival. I'm focusing on competitive interactions between species because there is a general agreement that negative interactions limit adaptation. For instance, competition can restrict the resource use, which leads to the decline in effective population size, lower supply of beneficial mutations, and lower adaptation rate. Also, there might be a trade-off between adaptation to abiotic and biotic component of environment. Recent studies suggest that evolutionary rescue would be also restricted by competition, especially in the circumstances where there is a strong niche overlap between species. However, some studies suggest that competition can promote adaptation. When conditions change, both species would be maladapted, and the average genotype of both species would lag behind the genotype optimal in new conditions. However, it is unlikely that both species would be equally maladapted. The species that starts closer to the moving optimum could benefit from competition because more maladapted species could increase the selective pressure, which would push the better adapted one towards the moving optimum. This hypothesis that has been developed recently has still not been experimentally tested. And sex is expected to be advantageous in these circumstances because it could generate the genotypes closer to the moving optimum and delay extinction as environment progressively deteriorates. Prolonged survival could also increase the likelihood for rising of beneficial mutations. I suggest that an interplay between competition and sex would promote evolutionary rescue of one species at the expense of another. I designed a selection experiment in order to address these questions. So can evolutionary rescue of a focal species be promoted by a competitor? And can sex and competition combined facilitate evolutionary rescue? Focal species used in this experiment was Chlamydomonas reinhardtii, which was divided into two groups of populations, sexual and asexual. Sexual populations had been allowed to undergo 3 rounds of sexual reproduction before being subjected to the lethal level of salt, along with the competitor, Chlamydomonas mevicii. After being mixed with a competitor, they were allowed to reproduce only mitotically, and asexual populations were reproducing mitotically throughout the whole course of experiment, before and after being mixed with Chlamydomonas mevicii. Mixed populations were propagated in liquid medium by serial transfers, and prior to each transfer, each population was sampled and plated on agar in order to assess population size by counting colonies. Each species was recorded as rescued if it scored the same or higher number of colonies each time it was sampled. Mixed populations were maintained until one species or both of them went extinct. So this is the general outline of experimental design. So we have mixed populations comprising both competitors, and if combination of sex and competition increases the likelihood for rescue, we should expect higher extinction rate when Reinhardtia reproduces asexually. We also have Reinhardtia populations propagated without a competitor, and if competition only promotes evolutionary rescue, we should expect higher extinction rate in these populations. And this experimental design had also allowed me to test for interactions between mode of reproduction and competition. And this is what I found. So this is the number of surviving populations, or the number of evolutionary rescue events of the focal species when propagated without a competitor. So as expected, based on the results of previous studies, this number was higher in sexual populations, so sex alone promoted evolutionary rescue. However, when competitor was added, this number declined irrespectively of mode of reproduction. So contrary to our hypothesis, competition reduced the chance for evolutionary rescue. The number of surviving species was higher in sexual populations, which indicates that sex mitigated the negative effects of competition and increased the likelihood for rescue even when competitor was present. Sex was advantageous both when competitor was present and when it was absent, so there was no interaction between mode of reproduction and competition. Sexual populations also had higher mean fitness compared to asexual populations. So here we can see the average number of cells per milliliter in sexual and asexual populations grown grown with or without competitor, and sexual populations had higher average population size in comparison with asexual populations. Addition of competitor led to decrease in population size for both mode of reproduction, but it was more prominent in sexual populations, whereas in asexual population that drop in population size wasn't that significant. So to sum up and give you some take-home messages, competition decreases the likelihood for evolutionary rescue. Sex increases the chance for evolutionary rescue irrespectively of the presence of competitor, even though that chance is higher when competitor was absent. And rescued sexual populations have higher mean fitness than asexual populations. And with this, I would like to finish and to thank my supervisor Nick Colgrave, all the past and present members of the lab, to my funding body, Darwin Trust of Edinburgh. Thank you for listening, and I'm happy to take any questions. Thank you.
Thank you very much. Yes, please.
Did you see any pattern to sort of the rate of extinction in sexuals versus asexuals? Are they going extinct sort of at different time points?
You mean the extinction dynamics?
That's right.
It was interesting because at first I observed higher extinction rates among asexual lines, but then it became reversed. And then sexual populations kind of leveled off and stopped being extinct, whereas asexual populations continued to go extinct until they leveled off as well. So that was interesting because it became reversed.
So when we know how do you equalize the initial condition for the sexual and asexual population with a recent environment?
How do you equalize the conditions for?
Yeah, the capability to survive in the same recent environment, asexual and sexual population is the same survival.
Yeah, so the only difference between them was their history of reproduction. So sexual populations had 3 rounds of sexual reproduction before being exposed to a lethal environment along with the competitor. And after that, sexual reproduction hasn't been induced. So they continue to reproduce asexually simply by means of cell division, simple cell division.
Yeah, so how about the survivors in the lethal environment? There are the same survivors between asexual and sexual genotypes, asexual and asexual populations. So if you consider evolutionary rescue, at the first point of the trend value should be equalized between sexual and asexual populations. So it is my comments. Okay. Maybe later.
I'm happy to talk about it later.
Okay.
Definitely. Yeah.
Okay, so thank you again very much.
Thank you.
And we come to our next speaker. I just have to find the presentation. But please come here. Which is Rachel Portals, is this correct?
Yes.
Okay, from Valencia.
Thank you very much for coming. I'm presenting a study I did with my colleague, Garcia Rocher. He couldn't come this time, but he says hello to all of you. Zooplankton organisms live in temporary habitats. This means that for a period of time, their habitat may become unavailable. This is obvious when the pond dries up, but it's also true in permanent waters due to the presence of antagonists or stressful physical conditions. So one way to escape is dormancy, mostly in the forms of diapausing eggs. So these diapausing eggs bridge growing seasons over a time of adversity. In many areas, especially in the Mediterranean, there is high unpredictability, so we don't know for how long these adverse periods or good condition periods are going to last. This is why when the habitat becomes available again, not all of the eggs hatch. Thus, diapause patterns can be characterized as the time an egg spends in the sediment or the fraction of the eggs that actually hatch in every hydro period. So the hatching fraction must be predicted, it's predicted to maximize fitness in the long term. So it has been suggested, so thank you, Nina, that in an unpredictable environment, only half of the eggs should hatch, while the other half should go through variable durations of diapause with hydro periods. And this has been described as a risk-spreading strategy, implying variable duration of diapause. However, the sediment is not out of risk, because there are also predators, antagonists, and stressful conditions. So the duration of the diapause is constrained by the probability of surviving in the sediments. This means that the uncertainty in the water column and the adversity of the sediment are expected to exert opposed selection pressures for diapause duration. That it's highly unpredictable, it would be selected very low hatching rates and prolonged diapause as long as the sediment is safe. On the contrary, if sediment adversity is increasing, we should expect higher hatching rates and shorter diapauses. So survival of the eggs and duration and hatching success are related to resource allocation. And we know that lipids play an important role in diapause of many zooplankton species. So diapausing eggs should contain enough resources to be maintained in the sediment, but also retain resources for post-diapause performance. Since these resources cannot be reallocated, trade-offs are expected to occur. So let me introduce you to my species of study, the rotifer Brachionus plicatilis. It's a monogonous rotifer inhabiting coastal ponds and saline lakes. They are very time-varying environments and temporary, so diapause is key in their life cycle. So the life cycle starts with a diapausing egg, resulting in asexual female, and then a phase of clonal propagation starts until population density induces sexual reproduction and the diapausing egg is formed again. These diapausing eggs have been described to survive for decades in the sediment. This is consistent with the idea that diapausing egg production is very costly. The rate of production of sexual eggs is much lower than the rate of production of asexual eggs. And also with the observation of lipid droplets in the diapausing egg. So we wanted to check the survival hatching rate and lipid content of diapausing eggs in populations, in cultures founded from eggs collected in the wild, test differences between these populations, and explore potential trade-offs. Our expectations were a negative association between survival in the sediment and hatching at the population level. So those populations with risky sediments should have higher hatching rates. And a positive association between diapausing egg survival and lipid content, thinking that lipids should maintain extended diapause through slow metabolism. We collected eggs from dried sediments of 9 ponds in Spain, and these ponds have been characterized as having different levels of predictability. We hatched the eggs, produced clones, combined these clones, so we ended up with lots of diapausing eggs, highly genetically variable for the 9 populations, and we made several batches for each population. With the first batch, we test for egg deterioration. So we kept those eggs for more than a year in the dark and wet conditions outdoors. And then every month, very carefully, we would observe the eggs and classify them according to the percentage of occupancy of the embryo in the eggshell. So you can see here just egg type 1 and 2 are going to make it. So they are the only viable eggs. With this data, we obtained the deterioration rate and the fraction of viable eggs. With the second batch, we did a hatching experiment. So after 1 month in the previous conditions, we exposed type 1 eggs to hatching conditions. And then we check weekly how many eggs had hatched, and we did this twice. And we obtained hatching dynamics and the hatching fraction. And a third batch was used to estimate the lipid content in the eggs. To do so, we encapsulated the eggs with bleach, and we stained them with oil red solution. And after some image treatment under the microscope, we measure lipid content as the mean pixel intensity of the red droplets. I don't know if you can see it, but high lipid content, low lipid content. Okay, so these are the results for the 9 populations of the deterioration rates. As you can see, after more than a year, most of the eggs, so more than 50% of the eggs were type 1 or type 2, so they maintain viability. There were differences, though, between populations. This was barely untouched, and this quickly deteriorated. And also the deterioration rate decreased with time. There were also differences between the hatching fraction from 5 to 52%, and also in the time of hatching, and also differences among populations in the lipid content. So there was a smooth gradient, and one population clearly differentiated. This population was the most predictable one, but when we compare with our predictable and unpredictable ponds, we didn't really find a relationship, neither with the other 2 traits that I showed you. Let's check the trade-offs. So this is the percentage of viable eggs, so eggs from type 1 and type 2, against the hatching fraction. We found a significant negative correlation, meaning that those populations with a low possibility to survive in the sediment would hatch earlier, just as we predicted. This means that risky sediments increased hatching rates. We don't know how risky our sediments are, but we have evidence that eggs in some of these ponds are deteriorating really quickly. When we check the survival against the lipid content, remember we expected more survival with more lipids, and we found exactly the contrary, a significant negative relationship. Those populations with less lipid content survived longer. Describe the drawbacks of lipid storage, for example, disrupting cell components, and thus increasing mortality. But if this is decreasing fitness, why are lipid droplets maintained? One possibility is that they are being retained for the performance of post-diapausing females. And in fact, post-diapausing females contain huge amounts of lipid droplets. Another possibility is that eggs are using these lipid droplets as a mechanism for buoyancy, and thus wind or animal dispersal. And this actually makes a lot of sense, because if the sediments are risky, they don't want to sink. They prefer to swim and get carried away to other ponds. If that's the case, if dispersal is mediated by these lipids, we should expect populations with higher amounts of lipids to hatch earlier, because for colonization to be successful, eggs must hatch really fast and then grow clonally. And this is exactly what we found in our results. So populations with higher lipid hatch earlier. In summary, eggs do deteriorate, but they do so differently among populations. They differ in survival, hatching fraction, and lipid content, but this is not related to habitat predictability, or pond size, or egg size, in case you're wondering. The negative correlation we expected between survival and hatching was true, so risky sediments increase hatching. And the correlation between egg survival and lipid content was not true. So lipid content apparently is doing no good to the eggs. So in that case, maybe dispersal in time and in space are working as alternative risk spreading strategies. These strategies trade off in several diapausing egg traits, and the final result will depend on a combination of factors on the habitat characteristics, and I'm sure hydroperiod and predictability are playing an important role. For example, diapausing eggs living in ponds with risky sediments will go for space dispersal, and therefore they should have eggs with higher lipid content that hatch earlier but live shorter. And this has already been demonstrated in plant seeds. Thank you for listening.
Thank you very much, Raquel. Other questions? Yes, please.
Given you didn't find what you expected in your correlation with lipids, did you explore any other ways of measuring the lipid content within the cells?
There are other ways, but not in a rotifer resting egg, which is 100 micrometers most.
I wonder if you thought about looking at a gravimetric thing rather than like an actual image analysis you use. Is there any way of extracting the lipid from the cell?
You're asking me if there could be some artifact in the treatment of the image? Is that what you're asking?
Yeah, potentially. I just wondered if you looked at doing a comparison between an image analysis of lipids as opposed to an extraction from the actual protein.
Well, we had the help of an expert, so
I was just curious.
we used an analysis to remove noise.
Yeah.
We were careful in that. Yeah.
Are there more questions? Yes, please.
Do you find the same variability in the same pond? So maybe I missed it, but you're comparing different ponds with different risky situations. But do you find also in the same pond a similar strategy, meaning that maybe some eggs are supposed to hatch faster and others are hatch later on to have that hatching strategy even in the one population?
Yeah. There's diversity within populations, so these are mean average data. Populations are also genetically differentiated. But yeah, on average, but you can see the both responses in the same ponds, yes.
Okay, let's stop here. Thank you.
Thank you.
And we come to our next speaker of the session, who is André Manzer. Is this all Swiss today, or you also Swiss, no? Okay. Let's see. Sorry. Mm-hmm. Okay. Sorry.
No problem.
But No, there's something completely different. No, that's okay. Fine, sorry.
I'm happy with that. Well, hello everyone. So one of the cornerstones of behavioral ecology and probably ecology as a whole is the notion of individual organisms as fitness maximizing agents. And accordingly, we think of us or mice as harmonious integrated wholes, units. Yet this is surely just a simplification. So if you look at this one mouse here, it usually consists of a myriad of cells on the order of 10 to the 14, which themselves contain about 10 to the 4 genes. So actually what we're looking at here is a collective. And now, interestingly, most of these members of the collective contribute in most elaborate ways to come up with complex organisms like us in here or these mice. Otherwise, we wouldn't be sitting in here and listening to talks. Although that's only sometimes the case, and we find more and more examples of genes or members of this collective that don't contribute to the organism as a whole and the function as a whole. And what I've looked at in my PhD is a particular special chromosome in mice that has managed to spread throughout populations around the world despite being lethal, so it doesn't contribute at all. Actually, it's harmful. This peculiar chromosome is called T haplotype, and as I told you, it's lethal in homozygotes, so animals that are homozygote die in utero before they're even born. Now, normally, if you had a gene like that or a chromosome like that, natural selection would take care of that within some generations, and it would be counter-selected, so we would get rid of that problem. But that doesn't happen here. So what is going on here? So in heterozygote males, what we're looking at here is actually a sperm killer. So in heterozygote males, normally what we would have is he would produce equal proportions of T sperm and wild-type sperm, here represented in orange and gray respectively. Now the T haplotype produces a poison protein that destroys flagellar function in all sperm first of all, and then locally produces an antidote that recovers the function of all those sperm that are carrying the T haplotype. So in effect, when it comes to the race to get to the site of fertilization, T sperm do very well, while the wild-type sperm are actually left somewhere behind. So we end up in a situation where fertilization probability systematically deviates from what we know from Mendel's rules, and about 90% of T sperm are actually managed to fertilize the eggs. Now, what are the consequences of this for individual organisms? Now, let's look at a female. Here, maybe a quick reminder, genetics 101, a cross between two heterozygotes. So what you normally would expect here is this classical a quarter, a half, a quarter proportions in the offspring. Now, because we have this drive effect here, it means that many more TT homozygotes are produced. And this in itself wouldn't be a problem, but as I told you before, they're lethal. So a female that actually mates with a heterozygote male, the T carrier would lose up to 45% of the offspring, which is a huge fitness cost. So we would expect actually the female to come up with strategies to avoid these costs related to that evil drive gene. And one hypothesis how she could do this that people have been looking at in the lab in particular is that she could mate with several males. So the idea of this would be that she mates with a second male, say this would be then a wild-type male, and he would also contribute a certain amount to a certain amount of sperm. And then when it comes to the sperm race, all of his sperm would actually do pretty well, whereas because the T actually attacks some of the wild-type sperm within the T male, he would only have fewer sperm, and through that, she could actually avoid some of the litter losses associated with the T haplotype. And people have been measuring this actually in the lab and have found that this poison antidote system that I've showed you earlier doesn't work properly, so even more sperm are affected, and only about 12% of eggs usually are fertilized by T males in lab experiments. So just to run through sort of the basic logic of the polyandry and as a suppressor of gene drive argument. So we have sort of female mating behavior in the form of polyandry, so she mates with several males, that then facilitates sperm competition, and the idea would be that drive males are bad sperm competitors. So this could explain actually, if you look at population consequences of the T haplotype, we typically would expect that because of drive, the combination of drive and lethality, about 2/3 of the population would carry that gene. So we would expect sort of a stable polymorphism. This is not what people find. So empirically, we usually find much lower frequencies. And this is a puzzle that has sort of haunted evolutionary biologists for over 50 years. And we think that maybe through that additional effect of polyandry, we could explain this discrepancy between the monandry prediction and empirical observation. We could also then have a feedback, because it's beneficial for a female to be polyandrous because it helps her avoid these harmful genes. That could actually mean that polyandry could be selected as a consequence of that. So we wanted to look at whether there's selections on polyandry, and in order to have evolution, we would also require heritability. So as I said before, we have good data for this sort of this whole complex or this whole argument in the lab, and what we really wanted to do here is to test these ideas in the wild. So what we were asking are 4 questions really related to these 4 dimensions here. So the first question would be, are mice really polyandrous? So do they mate with several males? Second, are drive males really, do we find evidence for males that carry the drive gene being bad at sperm competition? Does drive explain the observed frequencies in the wild? And also, could that facilitate the evolution of polyandry as a whole? Which is sort of in itself a question in evolution, a long-standing question in evolutionary biology. So we looked at this in a population of house mice in Zurich. It's sort of a long-term project. This population has been started in 2003. It's open to migration, so mice can leave and enter. It's sort of a barn building, and they're provided with food and water, and we extensively monitor. They're provided also with nest boxes, so we regularly monitor reproduction. We genetically sample them, so we have a full pedigree. We know which individuals were fathers and mothers of offspring, and we also have a marker that is specific for the T haplotype, so we can actually look at the frequency of that particular drive gene. So for this study here, we looked at a period between 2006 and 2010 in which about 3,200 mice were born. So to just give you a brief overview, first of all, here you see the number over the time period, sort of the reproductive activity in the barn, as expressed as the number of offspring, and in red you see newborns in the population that were carrying that T haplotype and newborns that were carrying the wild type. And as you can see, throughout the period we had a significant fraction of the population actually carrying the T, allowing us to address related questions. Coming to our first question, are mice actually polyandrous? So here again, I show sort of litters that were born within that time period. And on the y-axis, you have, on one hand, the number of offspring as a function of how many sires they had, and then the frequency of polyandry. And as you can see here, in red, there's many litters that have more than one genetic father, and it actually goes up to 4 within one litter. Overall, in the about 1,000 litters that we looked at, about half of them had more than one father. So there's strong polyandry, or females regularly mate with more than one male. The second question is, are drive males actually really bad at sperm competition? So in this plot here, we look at the average number of sperm competitors the male encountered during his lifetime. So if that's 0, in all of his offspring that he sired, he didn't have to compete against another male. And if the number goes up, he had experienced more competition against other males. And we plot that against reproductive success. What we find is, first of all, as you would expect, with increased levels of competition, his reproductive success goes down. I forgot to say that the size of the bubbles is the number of reproductive events. So that goes from 1 to 1 male managed actually to have be successful in siring 35 litters. More importantly, what we find is a significant interaction with T genotype. So T males are more strongly affected by sperm competition than wild type males, which is exactly in line with our hypothesis. And this actually translates into a significant difference in overall lifetime reproductive success between drive-carrying and non-drive-carrying males. when we look at whether there's selection for females, so what we would expect here is sort of a positive correlation between her polyandry rate and the reproduction she manages. Oh, good. We find no evidence for that. So again, there is quite a considerable amount of reproductive skew within the population, but that is completely unrelated to whether she carries the drive or not. We also performed a generalized animal model based on the pedigree to see whether we find any evidence for heritability of multiple mating or her tendency to have multiple males or sire offspring from multiple males, and we don't find any evidence for either heritability nor we find any signs of systematic individual variation. What we do find is that there's certain environmental variables, such as the density in the population, that is positively correlated with the probability of her giving litters with more than one father. Overall, if you go back, does that now have repercussions on drive frequency in the population? So that's the plot I sort of showed you in the beginning. And what we find here, again, we have the monandry prediction, which predicts that sort of 2/3 of the population should carry the T. And if you look at the frequency, it first of all goes down and is always sort of systematically different and lower than based on that prediction, exactly in line with what other people find. And we think that actually this effect that I've shown you before of polyandry can actually explain that discrepancy. So polyandry as an explanation for that low frequency here. So to go back to our initial questions, are mice polyandrous? Yes, we find strong evidence for that. Are drive males bad at sperm competition? Yes, for the first time in the wild, and we're quite excited about that. We can also show that this is the case. Does polyandry explain observed frequencies in the population? We think that's most likely the case, although we don't find any evidence for selection nor for heritability on polyandry. So that feedback there seems not to be the case, at least in the population that we looked at. We're at the Ecological conference, so does that have any ecological relevance? So here, to illustrate that, I sort of plot the frequency of drive individuals in the population against the mean population fitness, or the productiveness of the population. If we start somewhere in the middle, if it was a Mendelian trait, it would go extinct immediately and we would be at the optimum for the population. That's sort of the magic of meiosis, that you end up with, at the point which is optimal for the population. Because we now have this drive, we have a significant amount of individuals in the population that actually result in lethal offspring being produced, and actually that would result in almost 20% at stable equilibrium, 20% of the offspring dying. So that's an important ecological variable, I think. And the argument here would be that if you add polyandry, you can again sort of go back to the optimum level. So overall, I hopefully have shown you that sort of unraveling the conflicts within an organism can be very insightful, and drive genes can actually have very profound effects or implications on reproduction, mating behavior, and survival. The argument here was that polyandry can purge natural populations from those harmful effects of drives, which can be ecologically relevant. And importantly, I think these are insights that would not be possible if you would start from the notion of organisms as the sole fitness maximizing agents. With that, I want to thank people that helped me with this work and you for your attention. Yes.
Do you have a sense of what the distribution of that T genotype is in nature? The reason I ask is you can imagine if it was sort of spread out evenly, then polyandry could kind of work everywhere, but if it's clumped, then polyandry might not be as successful. Do you have any sort of sense?
I mean, people have measured or have been trying to find them throughout the world, and they find it in 4 mice subspecies, and as far as I can tell, it's fairly evenly distributed. I'm not quite sure why you would think that if it's more clumped that polyandry would be more effective, but that's maybe something...
Maybe less effective.
Less effective. And why would that? Why would you...
Because mating with more individuals would just mean you would just expose yourself to that genotype.
Ah, yeah, okay. Ah, yeah. Yeah, that is true. So the polyandry is inherently frequency dependent. You're completely right. So if everyone in the population carries the T, then you're less likely to encounter a wild type individual that would actually give you the benefits of sperm competition. Yeah, you're right. I think we don't know enough about, in general, we don't know enough about natural there's, but as far as I can tell, there's, not the case. Yeah. Yes?
If it's in 4 subspecies, then presumably it was in the common ancestor of all of those, which does imply that it's been around for quite a long time.
Yes, over 3 million years, actually, yeah.
So, I mean, this is just me rambling rather than asking a question, so it's pretty impressive that it's
Yes.
there.
Yes, so I think because it's lethal, it cannot go to fixation, so the maximum frequency it can reach is that everyone is heterozygous in the population. So we think that because it drives, so it's very strong if it's at low frequency, but it can't go to fixation, that could actually make it very stable. And there's been some phylogenetic analysis that suggests that there has actually been, that the most recent common ancestor of the T haplotype is younger than the most recent common ancestor of all 4 mouse subspecies. So that suggests that this gene has actually swept through the population after sort of the split of the species. Yeah.
Okay, thank you very much. And with this, we come to our next speaker, who is Seth Baribeau. He just started very recently in Liverpool. And I have to find.
All right, thank you very much for the chance to speak to you. And I'm going to be talking about one sort of a cousin of a side project I did while doing my postdoc, also in Zurich, although down the road at the ETH. And if you come from sort of an evolutionary ecology background, you probably know that generally sex is a terrible idea and it has lots of problems with it, which is why it's been one of these really exciting topics for understanding evolutionary biology. And it's terrible in a lot of different ways. You have to do things like find and attract a mate. Here I'm mostly talking about in the context where there's actually copulation as opposed to like broadcast spawning, but some of it applies more generally to sexual reproduction as a whole. So you have to find and attract a mate, which many of us know is not always easy or straightforward. And other organisms go through very dramatic displays of this. I think my favorite case is this thing. I don't know, have people seen this? This is this lovely display that was found on the seafloor off of an island in Japan, and it was very mysterious, and nobody knew what it was. And then they filmed it was being made by male pufferfish. So they make this beautiful sort of mandala shape on the ground, and females will come up to it and sort of wiggle along into the middle, and if they like the artistry of the male, they'll deposit eggs in the sort of bullseye spot. But clearly, very time-consuming, energetically consuming activity. It helps if you don't die in the process of finding a mate, and in some species that's actually fairly common. This is a species from the genus Portia, a jumping spider from northern Australia, where immature females will mimic sexually mature females and then mate with any of the males that come hopeful for a mating opportunity. Don't get a sexually transmitted infection, and these are very common. For instance, this is a sexually transmitted, that's not what I intended to do. Transmitted disease of Silene. This is anther smut, which, yes, even plants can get sexually transmitted infections, and these are transmitted, they're vectored by pollinators. Go back, keep going. Broken it. Do you know what I'm supposed to do here, Livy? Press one button wrong.
Yeah, then nothing works. We will find out.
Why does it say file?
I don't know.
Did you get a mouse on there?
So we close it and then.
We can try. I'm just pressing buttons now.
Mm-hmm.
Don't work. Oh, wait. Okay. Okay, there we go. All right, so the sexually transmitted diseases. Then there's just the fact that sex itself can be damaging. So this is a real website, but I would recommend Googling it in private. And these are some of the sort of more dramatic examples of sort of harmful sex. So this is the penis of a bean weevil, which is covered with all these spines. There's the dagger-like penis of a bedbug, which uses traumatic insemination, so it actually punctures the abdomen of females. And these are heavily weaponized versions of the normal sexual reproductive apparatus. And the idea here is that this prevents multiple mating. And we've just heard about how multiple mating can be great from a female perspective, but clearly not always from a male perspective. And these sort of harmful adaptations can help reduce multiple mating. All right. Then there's sort of more classic arguments for why sex is a terrible idea. You have to give up half of your genome. This is the twofold cost of meiosis, where whatever you did that got you to the point where you can reproduce means your genome is pretty good. You then have to shake it all up with another genome and hope that that is also good. And you can be breaking up really good combinations. And then there's the numeric cost of sex, which is simply that it takes 2 individuals to make the same number as would 1 individual of asexuals. So they should always sweep through the population. Now, that's why sex and the evolution of and maintenance of sex has been one of these enduring questions in evolutionary biology. And there's some really fantastic theories for how sex evolved and is maintained. And I'm going to largely ignore those and focus on something completely different and less appreciated, and it sort of comes from the title of this talk. So, Marvin Gaye suggested in 1982 in his song Sexual Healing, and I'm going to go through this, oh baby, I'm hot just like an oven, suggesting you may have had a fever. I need some loving. And baby, I can't hold it much longer. It's getting stronger and stronger. This part could be gastrointestinal distress. When I get that feeling, I want sexual healing. Sexual healing, oh baby, makes me feel so fine. And clearly what he's getting at this point is that there's something about sex or mating that will cure him of his ills. And this hypothesis was largely ignored by the academic community. At least for a reasonable amount of time. So this came out in '82. Some years later, people did start looking at mating and how that influences immunity. And they largely found the opposite effect, that sex corrupts immunity. So this idea that having a mating event can potentially turn on the immune system or can suppress the immune system, but in both of those cases, it's harmful. So you're turning on your immune response in order to fight something that's not a disease and then making yourself susceptible to a real disease. Right, so I was working with bumblebees, and bumblebees, unlike those highly weaponized penises that you saw before, bumblebees only mate singly. Exceptions, but most species mate singly. The mating's not that bad, but there's still some evidence of damage. So this is a figure from a book showing a queen after mating, and you get these melanized nodules after reproduction, and that's a classic insect wound healing response, the melanization response. So at least the question, why is there any sort of harm at all in this context? Why do they bother to do this at all when they mate singly, and that should be it? There should be no reason to harm a female. There should be no opportunity for multiple mating. So I was interested in seeing whether or not mating induces an immune response or what that looks like. I mean, it's suggestive that there is some from this wounding data, or this figure at least. And what does it mean when they then encounter a real parasite? And to do so, I took queens and I took males, and I either gave them an opportunity to mate or no opportunity to mate, and looked at their immune response as measured by gene expression, and then infected them with a trypanosome, which is this creature here. This is Crithidea bombi. It's a trypanosome gut parasite. It's very common in the environment, especially when queens emerge, sort of at the end of summer. What I found was that, in fact, mating did reduce the infection intensity in my first experiment. So here we're using a fairly arbitrary cutoff of 1,000 cells based on sort of the distribution of the data when I was looking at it. Males tend to be more highly infected than females, but we knew that already, or queens. But that mating in both cases tend to reduce the infection intensity. We also saw a number of genes that are differentially expressed upon mating, including these antimicrobial peptides, which are quite highly expressed after mating, and punch and hopscotch, which are involved in the melanization pathway, and relish, which is a transcription factor in the IMD pathway, which leads to production of antimicrobial peptides. Now, I was wondering perhaps even a social response, because you put a queen with a male or you have a queen by herself. It could simply be exposure to another potential mating partner, and it could be sort of a prophylactic induction of the immune response. So I repeated the experiment with a mated or a frustrated conditions, where they were put together, but they were not allowed to mate. And this was done by applying wax to the male to seal his abdominal tergites at the very tip of his tail. So he would try and mate, but was not able to do so. I could take the wax off, and it works just like with people. So I had some very upset male bees with manzillions, which I did not use in this experiment for gene expression because they were looking very upset and maybe would not be representative of what would really happen. So from here on out, I'm just talking about queens. And found that frustrated queens we still have a reasonably small infection. This is just yes or no, did they become infected or not? Reasonably few of them get infected, but far more than if they were allowed to mate properly. I was a little nervous about this number, given that it wasn't a huge mating success. And in both of these previous sections, I used 20,000 cells. So I doubled the number of cells and saw a similar pattern. So here again, increasing the infection likelihood, but still a big difference. So the frustrated queens are far more likely to be infected than if they were allowed to mate properly. And here I also had some data about the time series, and we see that the mated individuals here in the red triangles have higher expression of a number of receptor genes. These turn on the immune system if they find a pathogen. A number of effectors. Here's this hymenoptase, and all of these are antimicrobial peptides. These are some melanization response genes. So these red ones are much more highly expressed of all these immune genes relative to the frustrated individuals here, somewhat appropriately presented as blue balls. So mating is able to induce an immune response, and it can result in protection. Even this is measured a week after mating, so it's protective for a while. So there is such a thing as sexual healing, but it seems to involve bumblebees, which I don't think was really on Marvin Gaye's mind at the time. And I think while I've kind of put this forward as a bit of a silly or funny example, there are some interesting consequences of this. First, queens are emerging at a time of year when there's lots of diseases, and so mating and some small amount of damage could be beneficial for that queen. This parasite is particularly harmful for queens, and it reduces her likelihood of surviving hibernation. So a boost in the immune system at mating could help protect her through to colony foundation in the following year. And it also gives us perhaps an insight into what early sexual conflict could look like. When we look at these highly developed systems where there's sexual conflict with these weaponized penises, those are very far down the path of sexual antagonistic coevolution. But here you have a situation where this minor amount of male harm seems to induce an immune response in the queen and is actually in both of their interests before it could be perhaps taken off in order to limit multiple mating opportunities. And so with that, I'll say thank you. And I also say, as Olivia mentioned, I'm new here. I have a PhD position, so tell your students if they're interested in, I will not give them a silly project like that. I save those for myself to embarrass myself. But so a much more sensible project for a graduate student, so please let them know. And with that, I'll be happy to take any questions.
Thank you, Seth. I'm sure there are questions. Oh, yes, to sing again. Yes, please.
Nice talk, Seth. I think interpreting this as adaptive is a bit dangerous because if the females need a certain degree of immune response to survive hibernation, they should be selected to have that immune response anyway.
Which in a way I think they do. So, I mean, it is much harder to infect the queens in the first instance. So I take your point. It is a bit tricky if you start saying it's necessarily adaptive, but I could see a situation like this potentially as a way to improve resistance for some period of time being adaptive.
One thing you could argue is that if mating induces a certain amount of immune response, then they're all going to mate.
Which they do.
So in that case,
Yeah.
they should have the optimal immune response that they need, including that boost from mating, which is kind of...
Yeah, you kind of get in a bit of a circular situation there. And they do all mate, because if they don't mate, they won't hibernate. So there is absolutely that element to it. Although it may also depend, I guess, when it arose, because they have not always necessarily been in areas where they do have to hibernate. So it depends on how ancient it is. I mean, it also comes to a general thing that, well, if one mating induces an immune response which is beneficial, well, why don't two? Because we know that there are all these benefits to polyandry. even in this system. And so there also, it's not always clear why they aren't able to just continue to mate, because clearly some species do it as well. So that's a bit of an open question.
Are there more questions with that? So would you have expected a similar pattern if the infection success was much higher?
I suspect so. So I mean, doubled the infectious dose, and it did increase, I think up to a certain point. I think if you really blast them with the parasite, no matter what, some very small number of them won't get infected. What I think is also interesting, at least from the first part, is that there was also a benefit for males, which is not something that you would necessarily expect, because I can't imagine where the wounding takes place. But whatever this activation is doing, it's activating a very systemic immune response as well. So it's protecting in the gut, not at the location of mating, which you might expect otherwise for sexually transmitted infections. I went completely off topic there, but I don't know.
Okay, thank you, Seth.
Sure.
And we come to our next speaker. And I hope I pronounce your name right, sorry. Yuya Tachiki.
Thank you.
Thank you.
Thank you very much for giving me such a great opportunity to give a talk in front of you. I'm Yuya Tachiki from Kyoto University, and also I belong to the Kyushu University, Japan. I'm going to talk about the evolution of flowering time in monocarpic clonal plants in front of the animal ecologists. And clonal plants can switch their reproductive mode from clonal to sexual reproduction. Asexual reproduction produces a semi-autonomous module, which is genetically identical with its donor. And sexual reproduction produces seeds after fertilization that are genetically different from parents. Each semi-autonomous module is called ramete, and the set of ramete produced from one seed is called genet. Today, I'm going to focus on bamboo, which is very familiar with the Asian people. We utilize bamboo in various ways, such as house and food and sometimes clothing. But actually, our knowledge about their unique life history is quite limited. Bamboo is a clonal monocarpic plant that undergoes mass flowering after a long-term clonal growth. The time to flowering after germination is thought to be most likely determined by endogenous mechanisms, but not affected by environmental factors. So this is a flower of bamboo, which is similar to rice, because bamboo and rice belong to the same family. The bare ground in this picture here was caused by the simultaneous flowering and the death, subsequent death of bamboo. So the flowering of bamboo impacts have a large impact on forest ecosystem. It is known that flowering interval ranges from 3 years to more than 100 years, depending on the species, and flowering interval tend to increase from southern to northern region within their natural distributions. Especially in Japan, it is known that bamboo madake wait 120 years until flowering. The question, how such the geographic tendency is formed in the evolutionary history, have attracted a lot of ecologists for a long time. Interestingly, as well as the geographic pattern in flowering intervals, it is also known that the rhizome branching pattern is also different between temperate and tropical region. Rhizome in temperate region is called leptomorph type, which is long and slender form, resulting in the sparse distribution of rametes. And these kind of species wait long time flowering. On the other hand, a bamboo in tropical region have a pachymorph typed rhizome, which is short and thick form, and resulting in the cramped distribution of genet. So we can easily detect which genet is which. But in the case of leptomorph type in temperate region, we could not detect from the above ground vegetation. So in this article, they suggest that there is some relationship between the rhizome system and the flowering interval. But what caused the evolutionary consequence of flowering interval is remain unsolved. I'm going to show you some pictures of the pachymorph on the right, on the left, and the leptomorph type bamboo in temperate region on the right. So our question in this work is as follows. What determines the flowering interval as a consequence of evolution? How rhizome system affects geographic tendency of flowering interval in bamboos? So we are exploring the evolution of flowering interval in bamboo in this talk. Our hypothesis is that the species with long rhizomes should have the longer flowering interval as a consequence of evolution, and vice versa. So to solve this problem, we adopt a two-dimensional lattice model in which the location of each ramет is recorded. In particular, age and trait value of ramет at each coordinate is assigned. We also record the genet ID, which genet each ramет belongs to. We simplify the life history of bamboo. After germination, bamboo continues clonal reproduction for tau years, which is genetically determined time until flowering. And then flower and die after seed dispersal. We assume that 4 events occur in each year. First, each ramет die with probability delta. Second, ramets whose age reaches tau flower disperses and die. These 2 events make free spaces by the death of ramets. And the following events, clonal reproduction and seed germination, fill the empty space. If empty space is located here, this space might be filled by the clonal offspring of ramets located at a particular range. So this distance depends on the length of rhizome. If it is long, the species is leptomorph type in temperate region. Otherwise, the species is pachymorph in tropical region. The probability that the free space is filled is mathematically described by the Poisson distribution like this. And the important parameter is alpha here. As alpha increases, the probability of establishment increases. So I will explain the process of sexual reproduction. If ramets flower, they exchange pollen with surrounding flowering ramets. When pollen comes from the same genet, the amount of seeds produced is reduced due to inbreeding depression. So here, HID indicates the strength of inbreeding depression. So we simply assume that the inbreeding depression reduces the number of seeds produced. The empty space is filled by seed germination as well as clonal offspring. The probability that seeds occupy the free space follows like this, here. And in beta in this function determines the probability to be filled the empty space. This is similar to alpha in the case of clonal growth. When empty space is filled clonally or sexually, new ramets inherit traits from the parent. When new ramet is produced clonally, new ramet has the same trait age, and genetic ID with its donor. On the other hand, if new ramete is produced by sexual reproduction, trait value tau is the same with one of the parent with 50-50 probability. This is the assumption. And the age of the ramete is reset to 0, and new genetic ID is allotted. Here we assume that mutation occurs with small probability. In this case, mutant has a trait value tau, which is the flowering interval, slightly different from its parent. So this causes the mutation and evolution. So repeating above processes, we can examine the evolution of flowering interval. So this movie indicates the population dynamics, and each color corresponds to the genetic ID. So the same color indicates the same individual. So when we start the simulation run at the different initial condition, the evolutionary trajectory of flowering interval converges to a particular value. In this parameter set, that we can conclude that the flowering interval, the evolutionary consequence of flowering interval is about 40 years. We systematically tested the impact of each parameter. For example, flowering interval is determined by the balance between the efficiencies of clonal growth, alpha, and sexual reproduction, beta. As alpha increased or as beta decreased, flowering interval increased as consequences of evolution. Now we test the impact of rhizome. As we expected, flowering interval increased with an increase in rhizome length. It is consistent with the observed geographic pattern in the wild. Why this tendency is caused is explained in terms of the spatial distribution of ramete. When rhizome is short, genet make a structure like this. On the other hand, if rhizome is long, each genet spatially intermingled with each other. Such spatial distribution mediates the degree of intragenet competition, which is probability that rametes belonging to the same genet competes over the same empty space. You can see that the species with short rhizome here suffers from the severe intragenet competition. This intragenet competition should reduce the efficiency of clonal growth, resulting in short flowering interval. So we also tested the effects of inbreeding depression. You can see that the flowering interval increased as the strength of inbreeding depression increased. So this is caused simply by the reduction of seeds produced. So this reduces the efficiency of sexual reproduction. So now I'd like to summarize the result of evolutionary simulation. Flowering interval was determined by the balance between the efficiencies of clonal growth and sexual reproduction. Long rhizome promoted the delayed flowering. Inbreeding depression also promoted the delayed flowering. So these kind of things are caused by the intragenet competition. So our mathematical model demonstrated that the difference in rhizome system remarkably impacted on the evolutionary consequence of flowering interval in bamboo. Considering with the fact that species with shorter rhizome have higher chance of inbreeding, for such species, inbreeding depression can easily be purged by the natural selection. Hence, the combination of rhizome length and inbreeding depression should contribute to the formation of geographic trend in the flowering interval in bamboo. So this work was done by the collaboration with empirical ecologists and supported by the Japanese Society for Promoting Sciences. And I'd like to thank to editor and staff of Journal of Ecology for Harper Prize and giving me such a great opportunity to talk in front of you. Thank you very much.
interesting your model contained nothing about seed predation, which is often attributed as a cause of delayed Paris Big Bang reproduction. Do you have a comment on that?
Oh yes, there is one interesting moth to eat the seed of bamboo. So they have a very, very long diapause. And so maybe it may work. But for the other seed predators, such as rodents and birds, still exist for a long time, and the number of such a seed predator depends on the other food resources than bamboo. So because bamboo does not flower for a long time. Yeah.
Any more questions?
So do you have an explanation for the geographic gradient and for the root length?
Yeah, so I have two hypotheses to form a difference in rhizome length. One is simply the maintenance cost, because in tropical region there is a high activity of soil fungi and other decomposers. So maybe it have a cost to maintain longer rhizome. And another one is the king competition. So to avoid the king competition, or the spread rapidly, longer rhizome is adaptive. Okay.
Yeah, but why would that be then this way? Wouldn't you then expect them to disperse more in more higher latitudes? Like,
Yeah, I think there's a spatial heterogeneity. So the fine-scaled spatial heterogeneity may work to maintain the shorter rhizome in tropical region. I think in the tropical region there is a really dark and some very few number of gap site is formed because in tropical region there is no big large-scale disturbances. On the other hand, in temperate region, there is a seasonal forest, and there have some big disturbances such as typhoon or landslides. So the spatial difference in spatial heterogeneity mediates the how long rhizome evolves. Or another hypothesis is simply the maintenance cost.
Thank you very much. And we come to our last speaker of the session, who is Robert Laird.
Thank you. Well, thank you very much. I've really enjoyed all these talks on sexual reproduction. I'm going to be talking about a species that is allegedly a flowering plant, but in my entire time working on it, I've never actually seen a flower yet. So a little bit of a difference compared to the other ones so far. What I'll primarily be talking about is the evolution of senescence. So senescence is the progressive physiological deterioration that occurs at the individual level. And this produces at the population level reductions in survivorship and also reductions in reproduction with increasing chronological age. And those two things are typically thought of as the two main components of senescence. And I'll be talking today about parental age effects, looking at what really could be considered a third component of senescence. So I won't dwell too long on evolutionary theories of senescence, but all of them, despite their subtle differences and distinctions between them, they're all basically predicated on the idea that the strength of selection is going to be waning with advancing chronological age. And some of the reasons for this are things like the idea that individuals are going to die for non-senescence reasons. So individuals will die due to disturbance or predation or communicable diseases and so forth. So the potential for immortality is not there. There's always going to be fewer individuals in older age classes on some level, and therefore we expect to see reduced selection on those older age classes. And this also goes along with the idea of the fact that the residual reproductive value should be declining with age as well as the compound interest problem, right? So we have this premium on early reproduction as well. Now, the thing about all of these types of parts of these evolutionary theories of senescence is just as they apply to survival and reproduction, declining with advancing age, we might also expect them to see declines in offspring quality with increasing parental age. And that's what I'm looking at in this study. So in particular with offspring quality, one thing that's already been investigated quite a lot and has been found in many different taxa is what's known as the Lansing effect. And it's a decrease in lifespan with advancing parental age. And it's been shown, the name of the Lansing effect came from a paper that was by Lansing, obviously, on rotifers. It's also been shown in ladybird beetles as another example, and in a whole host of other species ranging from duckweeds, which is the focus of this study, to humans. And in fact, probably the first study on parental age effects was actually on humans and was performed, interestingly, by Alexander Graham Bell in 1918. So this is the inventor of the telephone, but also apparently was one of the early workers on parental age effects in demography, which I found fascinating, but kind of an aside. So what you see here, though, are data showing that average offspring longevity on the vertical axis is negatively related to maternal age class. So offspring of older mothers tended to have shorter average longevity compared to those of younger mothers. And the same actually applied to paternal age class as well. So we know that offspring lifespan tends to decrease with increasing parental age. The part of the issue with this is that lifespan is generally a poor proxy for fitness. And in particular, this is because it doesn't account for the degree and timing of reproductive output, which are very important in terms of fitness. So if we really want to document this third component of senescence, we need to look at something that's going to not just... Comes back? Okay. It did indeed. Okay. But also something that accounts for this timing of reproduction. And so what we used in this was the intrinsic rate of increase, R, the dominant eigenvalue of a Leslie matrix. The study species that we used in this study was Lemna minor, or common duckweed. It's found quite cosmopolitanly, if that's a word, around the world. As I say, it's one of the smallest flowering plants. Very, very tiny, about 5 square millimeters in the strains that we've been dealing with. It's a very handsome species. It's often used to sort of spruce up depictions of less charismatic taxa, as you can see here. It has some real advantages for this kind of demographic work, which requires longitudinal and often multi-generational data collection. So as I mentioned, it's very small. It's short-lived, so they live, as if they're first daughters, that is, they live about a month. And we already know certain things that decrease with increasing parental age. So offspring size certainly does. Lifespan has been found, so the Lansing effect, as I mentioned earlier, has been found in this species as well as total lifetime reproductive output. So in this study, there were basically 2 phases. The phase 1 was for looking at these first 2 components of reproduction, where we followed 216 focal fronds from detachment, which we defined as birth, to death, which we defined as the detachment of their last offspring. And so from this, we could get survival and reproduction data after daily views of what was happening with these individuals. And then in phase 2, we started with 41 parental individuals that cumulatively had 542 daughter fronds. And from this, we were able to look at parental age effects. So fitness, as in the intrinsic rate of natural increase, some ancillary fitness measurements that I'll get into, as well as some shape indices that I'll also mention later. So I hope this is going to fit on here. It did indeed. Okay, so what we see here are on the first 2 panels, the left panel and the middle panel, are what you could call these first 2 components of senescence. So in terms of the proportion surviving, you can see that, I don't know, okay, so at the beginning here, up till about 20 days, basically all individuals survived, And following that, there was a very rapid decrease in survivorship. And in addition to this, the proportion reproducing also fell off over the course of lifetime. So this is the traditional measures of senescence. When we look at offspring fitness, though, we also see that as the parental age increased, the offspring fitness dropped off quite a lot. So the offspring quality, in terms of a relevant fitness measurement, declined quite strongly with increasing parental age being a potential third component of senescence here. In terms of ancillary measures of fitness, total offspring also decreased a lot, especially with those that were born or that detached from parents that were older than 20 days at their time of detachment. The latency to reproduce, probably even more important in terms of its effect on fitness and on the intrinsic rate of increase, increased quite a lot with advancing parental age, so it took longer for the first offspring to detach when they were of an older parent. And surprisingly, there wasn't actually much evidence for the Lansing effect at all in this study. So there may at the very end be a bit of a downturn in the lifespan of offspring with advancing parental age. But this one's really the most important one in terms of offspring fitness. So what this might suggest is that if we're going to model age structure in populations, we might not only need to model age classes of individuals, so what age an actual individual has in terms of its own survival and reproduction, but we may need to structure populations in order to model them correctly under these circumstances by both age and parental age. And the way we do this is basically by extending these population vectors, so this is for a population that only has 3 age classes, so that we have these blocks that represent parental age classes, and then the actual age class of the individual nested within the age class of its parent. And this complicates things in terms of the Leslie matrix as well. So if we look at a traditional Leslie matrix with fecundity shown in blue and survival shown in red, it's going to look something like this. And if we have these strong parental age effects, it suggests we might need an extended Leslie matrix approach, where again we have these blocks of matrices that represent parental age class, and then nested within them are the age classes of the focal individuals themselves. And I won't get into it too much today, but comparing the traditional Leslie matrix approach with this extended Leslie matrix approach, it does show that the sensitivity to selection using the extended Leslie matrix approach falls more rapidly compared to the sensitivity under the Leslie matrix approach with advancing age. So it does suggest that this really is a third component of senescence, although how important it is still something we're working on. So the final thing I'll look at is something that we've done more recently, and that's looking at size and shape measures using this phase 2 of our data. And so what we looked at were the effects of parental age on offspring area, on continuous symmetry measure, which is a measure of bilateral symmetry that's basically shows how far the shape of an outline of an individual is to the closest bilaterally symmetrical shape, as well as the circularity of the individuals. And these data were collected late in life when the individuals had reached their full size and after all of their offspring, effectively the grand offspring of the original progenitor individuals, were already detached. So as with other studies on aging in lemna, we found that the offspring of older parents were much, much smaller than the offspring of younger parents. So you can see it declines from somewhere around 7 square millimeters of very young parents to almost around 1 square millimeter amongst very old parents. In order to get at our continuous symmetry and circularity measures, we started with images of all the lemnifrons, and again, it's late in life once all their daughters are detached. We were able to turn this into a series of landmarks, 200 landmarks around the outside of the individual, and then calculate continuous symmetry by contrasting the actual outline of the shape shown as the black outline here with the closest bilateral shape. And it's a bit difficult to see because this particular frond was quite bilaterally symmetrical. But the the underlying gray shape is the closest bilaterally symmetrical shape in this case. And so the index measures the total cumulative distance between those two. So we expected that symmetry would actually increase, or would decrease rather, with parental age. But we didn't really see much evidence of that. So here we have the natural log of continuous symmetry on the y-axis. And so ones that are more symmetrical have lower values, and ones that have higher values are less symmetrical. And there actually wasn't that great of a difference there. However, we did note that the offspring of later individuals or older individuals were more circular. And this may have to do with why a bilateral symmetry didn't seem to increase at all, or didn't seem to decrease at all. So to summarize this, senescence really does have three components in lemna minor: survival, reproduction, and offspring fitness. So it may be important to include parental age when we're looking at age-structured models of populations. And in terms of shape, the offspring of older parents do show shape differences compared to those of younger parents, although it doesn't seem to be visible in terms of bilateral symmetry as we expected based on preliminary data. Okay, thanks very much.
Thank you.
Thanks.
Question, Robert? Yes, please.
Particularly in this clonal senescence, because what you say is that the individual clones, the rabbits are senescent. But if this was a tree, these would be the leaves, and leaves senesce.
Right.
Trees live a very long time.
Okay.
I'm just not sure that you're talking about
It's
senescence in the sense we normally apply it to an individual genet.
right. So the genet lives on because the individuals keep reproducing. I think they're a bit different than leaves because they themselves release daughters as well. So it's definitely clonal senescence rather than, or senescence of the ramete rather than senescence of the genet as a whole. What I'd like to do is to take a population, and this is a plan actually, and observe senescence now and then let it continue to reproduce vegetatively and do the same thing in a year and 2 years and so forth to see if there's actually evidence of senescence at the entire genet level as well. But you're absolutely right that this is ramete senescence.
Okay, so I want to thank all speakers for the session, and I hope that we continue doing the coffee break if we can. Thanks. Thank you.